Welcome to the class. Today we are going to start plant transmission method, mainly the agrobacterium mediated transformation. Transformation is defined as a process in which genetic material of an organism is altered by the integration of new genes into its genome. Transformation is done by using vectors. There are several vectors which are used like plasmids, cosmids, plasmids, etc. The importance of this transformation is to produce transgenic plant for having improved crop yields, improvement of different varietal traits, and transgenic plants have protection against parasites, pests, and harsh weather conditions. Transgenic plants are plants that have their genomes modified through genetic engineering techniques, either by the addition of a foreign gene or removal of a certain detrimental gene, any toxic gene. A foreign gene inserted into a plant can be of different species or even kingdom. The first transgenic plant was developed through the insertion of NPT2, that is neomycin phosphotransferase 2 bacterial antibiotic resistance gene into tobacco. There are several applications of transgenic plants in agriculture. First is resistance to biotic stresses. Biotic stresses are caused to the plants by infection of bacteria, viruses, by grazing of herbivores, or by some weed plants which grow in the fields. If we are engineering resistance to biotic stress in crop plants, it will help us to reduce the usage of expensive pesticides and herbicides in agriculture, and it will lead to a greater yield and better quality of crops. And it will benefit the environment also, as we are not using pesticides and herbicide in higher amount now after the usage of transgenic plants. Bacillus thuringiensis has been inserted in Dora plants for having insect resistance. Such crops are known as Bt crops, and these are an example of transgenic plant produced through this method. In addition, virus resistant plants can be achieved through the introduction of viral coat protein into plants. In images, you can see here the comparison of Bt crops with the non-Bt crops. So these are the Bt crops which you can see here in the circle. This is a Bt brinjal. This one is a Bt corn. This is non-Bt. This is also non-Bt brinjal. And this is Bt cotton, which is having healthy cotton, and leaves are also healthy. So you can see the effect of insect attack on non-Bt crops, whereas the Bt crops are not having any such remarkable effect of insect attack on their fruits and leaves. The second application of transgenic crops is resistance to abiotic stress. Abiotic stress are caused by changes to soil composition, humidity, water, sunlight availability, etc. Transgenic, which are having abiotic stresses, have been generated by the manipulation of transcription factors, late embryogenesis abundant proteins, which are known as LEA proteins, and antioxidant proteins, which have been successfully produced in plants and make plants tolerant to drought and salinity. Overexpression of the proline biosynthesis enzyme P5C, which allows the accumulation of osmoprotectant during drought season, provides transgenic plants with osmotic stress resistance. Osmoprotectants are such chemicals which are produced by the plants, and they render the osmotic resistance to the plant in stress condition. For example, proline, glycine, betaine, mannitol, sorbitol are some such osmoprotectants. Third application of these transgenic crop is having improving crop yield and nutritional value. So this technique is known as biofortification. Biofortification is a technique which is used in agriculture to increase the nutritional value of a crop. A well-known example, a very famous example would be the golden rice, a variety of Rhiza sativa which is produced to biosynthesize beta-carotene through genetic modification. The rice crop does not produce beta-carotene, but it has a precursor which is needed to produce beta-carotene. So the golden rice was developed by adding two beta-carotene synthesis genes. One is phytoene synthase (PSY) gene and one is lycopene beta cyclase (beta-SCY) gene, which has been originated from a plant, that is daffodil plant. And here you can see the comparison of normal rice and golden rice. The golden rice has yellowish color due to the production of beta carotene. And if beta carotene is present in your diet, your body can make vitamin A from it. That's why this is known as golden A rice, which is rich in vitamin A. Fourth application of transgenic plants is bioreactors. Bioreactors for recombinant proteins, including antibodies, metabolites, catabolites, proteins, and vaccines. Antibodies and vaccines against gastrointestinal tract disease, which are also known as GI disease, cholera, malaria, are known to be produced in transgenic plants such as potato, banana, algae, and tobacco also. An anti-cancer antibody that recognizes cell of lung, breast, and colon cancer had also been successfully expressed in rice and wheat seeds. The only plant produced Newcastle disease vaccine had been approved by United States Department of Agriculture for poultry farming with several vaccines in clinical trials. For making transgenic plants, you will need a gene construct. Simple functional gene construct consists of a promoter region, a gene coding region, and a terminator or stop region. In addition, certain gene constructs may contain special sequences such as enhancers, silencer, or reporter sequences depending on the nature of study. These are left borders and right borders of the gene construct. This is having a promoter region, mostly in dicot plant. The promoter used is CAMB35S, which is taken from cauliflower mosaic virus. This is a selectable marker. This is antibiotic resistance gene, that is HPT2 hygromycin phosphotransferase. This is terminator sequence. This is the gene of interest. Here it is shown GFP gene that encodes for green fluorescent protein. And these are several sites for restriction enzymes to cut. Transformation was the first time applied by Frederick Griffith in 1928 in Diplococcus pneumoniae, and transformation using electroporation was developed in late 1980s. Particle bombardment, that is popularly known as gene gun method, was also given by John Sanford in 80s. Agrobacterium tumefaciens or agrobacterium mediated gene transfer. Agrobacterium is a soil-borne gram-negative bacterium. It is rod-shaped and motile and belongs to the bacterial family of Rhizobiaceae. Agrobacterium tumefaciens is a phytopathogen and it is treated as a nature's most effective plant genetic engineer. There are three species of agrobacterium. One is Agrobacterium tumefaciens, which causes ground gall diseases in plants. The other one is Agrobacterium rhizogenes, which causes hairy root disease. And one is Agrobacterium radiobacter, which is a virulent strain. Here in this picture, you can see a large crown gall developing on a plant, which is caused by the infection of Agrobacterium tumefaciens. Crown gall disease and TI plasmid. Almost 100 years ago, Smith and Townsend postulated that the bacterium was the causative agent of crown gall tumors, although its importance was recognized much later. As Agrobacterium tumefaciens infects wounded or damaged plant tissue, it induces the formation of a plant tumor called crown gall. Entry of the bacterium into the plant tissue is facilitated by the release of certain phenolic compounds like acetylsyrenzone or hydroxyacetylsyrenzone by the wounded sites of the plant. Organization of TI plasmid. TI plasmid is approximate size as 200 kb, exists as independent replicating circular DNA molecules within the agrobacterium cells. The tDNA region, that is known as transferred DNA, it is variable in length in the range of 12 to 24 kb, which depends on the bacterial strain from which this TI plasmid has come. Nopalene strain of TI plasmid have one tDNA with length of 20 kb, while octopene strains have two DNA regions referred to as TL and TR that are respectively 14 kb and 7 kb in length. This TI plasmid, this is origin, that is the origin of the application. These are opine catabolism gene. This is which region? Genes which are having virulence, where genes. This is left border and this is right border. Anything between left border and right border is known as tDNA region. And it mainly contains genes for hormone synthesis, auxin and cytokinin, and genes for opine synthesis. tDNA region has the genes for the biosynthesis of auxin, cytokinin, and opines, and is flanked by the left and right border. These three genes, OX, Cyto, and OCS, are referred to as oncogenes as they are the determinants of the tumor phenotype in the plants. tDNA borders, that is a set of 24 kb sequences present on either side, right and left side of tDNA, are also transferred to the plant cell. It is now clearly established that the right border is more critical to DNA transfer and tumorigenesis. Virulence region, genes responsible for the transfer of tDNA into the host plant are located outside tDNA. And the region is referred to as Vir or virulence region. Vir region codes for proteins which are involved in tDNA transfer. At least 9 Vir genes or proteins have been identified. This includes Vir A, Vir G, Vir B1, Vir C1, Vir D1, D2, and D4, and Vir E1 and E2. Next is opine catabolism region. This region codes for proteins involved in the uptake and metabolism of opines. Besides the above three, there is uri region that is responsible for the origin of DNA replication and permits the TI plasmid to be stably maintained in Agrobacterium tumefaciens. Procedure of tDNA transfer and integration. So first, it will require signal transduction to Agrobacterium. The wounded plant cells release certain chemicals, phenolic compounds, and sugars which are recognized as signals by Agrobacterium. Next is production of tDNA strand. The right and left borders of tDNA are recognized by their D1 or their D2 proteins. These proteins are involved in the production of single-stranded tDNA. Its protection and export to plant cell. Single-stranded tDNA gets attached to with D2. Transfer of tDNA into agrobacterium. The single-stranded tDNA with D2 complex in association with Vir-G is exported from the bacterial cells. Vir-B products form the transport apparatus. Next is transfer of tDNA into plant cell and final integration. tDNA with D2 complex crosses the plant plasma membrane. In the plant cells, this tDNA gets covered with Vir-E2, and this covering protects the tDNA from degradation by nucleases. Where D2 and where E2 interacts with a variety of plant proteins which influences tDNA transport and integration. The tDNA where D2 and where E2 plant protein complex enters the nucleus through nuclear pore complex. Within the nucleus, the tDNA gets integrated into the plant chromosome through a process which is recombination. This is different from the homologous recombination as it does not depend upon the sequence similarity. Agrobacterium rhizogenes can infect plants. It results in hairy root and it does not causing crown galls as in the case of Agrobacterium tumefaciens. The plasmids of Agrobacterium rhizogenes have been isolated and characterized. These plasmids referred to as RI plasmids. RI stands for root-inducing plasmids. These are of different types. Some of the RI plasmid strains possess genes that are homologous to TI plasmid, for example, auxin biosynthetic genes. Instead of virulence genes, RI plasmid contain a series of open reading frames on the tDNA. The product of these genes are involved in the metabolism of plant growth regulators which get sensitized to auxin and lead to root formation. The DNA carriers are the vectors which must have origin replication, a selectable marker, mostly an antibiotic resistance gene, and it should give permission to host to grow on selective media. Increase the volume of vector in the host cell. Multiple cloning sites are mandatory, and it allows insertion into foreign DNA. In this process, TI plasmids serve as natural vectors. However, there are several limitations to the use TI plasmid directly as cloning vectors. TI plasmids are large in size, around 200 to 800 kb. Smaller vectors are preferred for recombinant experiments. For this reason, large segments of DNA of TI plasmid not essential for cloning and they must be removed. Absence of unique restriction enzyme sites on TI plasmid is not preferable for transformation. The phytohormones, auxin and cytokinin produced prevent the plant cells being regenerated into plants. Therefore, auxin and cytokinin genes must be removed. Opine production in transformed plant cells lowers the plant yield. Therefore, opine synthesis First is factors. They are mainly composed of following components. First, the right border sequence of tDNA, which is absolutely required for tDNA integration into plant cell DNA. A multiple cloning site that promotes the insertion of cloned DNA into the region between tDNA borders, and an origin of DNA replication that allows the plasmid to multiply in E. coli. A selectable marker gene, NPT2, HPT2 in E. coli, and there are two types of TI plasmid derived vectors which are used for genetic transformation of plants: co-integrate vectors and binary vectors. This is a co-integrate vector system. In the co-integrate vector system, the disarmed and modified TI plasmid combines with an intermediate cloning vector to produce a recombinant TI plasmid. This is a disarmed TI plasmid from where the hormone biosynthetic genes and opine synthesizing genes have been removed, and then it is cloned with the intermediate vector. Production of disarmed TI plasmid. The tDNA genes for hormone biosynthesis are removed. So this is the left border and this is the right border. Between these is the tDNA region. So what has been done? This auxin and cytokinin synthesizing genes has been removed. And here, in the place of this removed sequence, the bacterial plasmid DNA sequence is incorporated. This Trian plasmid, also referred to as septal plasmid, has the basic structure of tDNA necessary to transfer the plant cells basically. This is the primary basic structure of this TI plasmid with origin of replication, opine catabolism genes, virulence genes, opine synthesizing genes, and auxin and cytokines. The intermediate vector is constructed with the following components. First, a PBR322 sequence, DNA homologous to this found in the receptor TI plasmid, a plant transformation marker which has been shown by the words PTM, a gene coding for neomycin phosphotransferase 2 NPT2. This lactobacterium cells with the receptor TI plasmid and immediate vector are selectively isolated when grown on a minimal media containing spectinomycin. The selection process becomes easy since E. coli does not grow on a minimal medium in which agrobacterium grows. Within the agrobacterium cells, intermediate plasmid gets integrated into the receptor Ti plasmid to produce co-integrate plasmid. This plasmid containing plant transformation marker, for example, neomycin phosphotransferase 2 gene (NPT2 gene) and cloned target gene between tDNA borders is transferred into plant cells. Transformed plant cells can be selected on a medium containing kanamycin when the plant and agrobacterium cells are incubated together. These are the advantages of co-integrate vectors. Target gene can be easily cloned, the plasmid is relatively small, and intermediate plasmid is conveniently cloned in E. coli and transferred to agrobacterium. So that was all for the day from this slide, binary vectors, and further we will do in the next class. Thank you so much for listening carefully.